My name is Spencer Schwarz. I'm a product manager for Canopy Biosciences, and I specialize in the instrumentation that we sell and the software that supports it. So today we'll talk about ChipCytometry. So this is a spatial biology platform, and we'll kind of get into what that means and who is and who isn't spatial and all sorts of stuff like that, because maybe got a little bit of a hot take on that too, which is fun. But kind of what we'll go through anyways is first, what is it in broad strokes, and then kind of getting into some of the details and some of the things that you can do with it. So that's the idea. Okay. Let's see if this still works. Nope. There we go. Okay, so ChipCytometry is a cyclic immunofluorescence staining protocol, essentially, in its essence. So essentially what we're going to do is we're going to take your piece of tissue, doesn't really matter what it is, as long as you can section it or you can pipette it onto a cover slip. It's something that we can use. And we're going to go through rounds of iterative staining with commercially available third-party antigens. And really what we mean by this is, anything you can get from BD or BioLegend, anything that you're already using for flow or for IHC, that's something that we can use. So we don't really have any proprietary reagents or anything like that. We'll kind of get through some of the consequences and the good stuff that comes out of this. But again, so kind of what we're going to do here is we're going to cycle through. Excellent. And 5 at a time, we're going to add stains to your tissue and we're going to collate those together. So in this case, this is fairly modest. This is 15 markers on a piece of colorectal cancer, but we can do 30, we can do 45 and things like that. Essentially, we haven't necessarily found the real limit to how many cycles you can do. That's going to really be more up to your tissue that we're looking at as opposed to the process itself. The next half of this, after we've collected the data, is we are going to use cell segmentation and a DAPI signal in order to identify individual cells. Then we're going to collate those signals. We have a very nice kind of quantitative way of collating signals. We're going to collate each of the signals that you've identified through your staining, and then we're going to cast that at something that looks very much like a dot plot in flow cytometry. So then the analytical process, or at least the one that's on board, is a process of defining phenotypes and subphenotypes based on gating, so that in the end, what you end up with is an identification of the cells that actually have that particular phenotype. So essentially, kind of everything in the middle there is kind of up to you as far as coming up with these assays. So what we're trying to do is basically use a microscope to emulate what you can do with a flow cytometer. And again, in the end, really what we have is a comparison between pieces of tissue, regions of tissue, or alternatively, if we're going to do a cell suspension, we could also look at differences in different treatments, whatever you happen to be doing. So that's the general idea. So we're going to use cyclic staining in order to add all these stains onto your piece of tissue, on your cell suspension, and then we're going to essentially use image analysis in order to define those individual populations. So there's some other stuff that kind of goes into this as well. Sorry, one more kind of aside here just to kind of bring it home. Again, we have because we found each of these individual cells, we actually have now a location in space and an identification of each and every one of these cells that are in our sample. So for instance, I know that, in that top cell, the guy in the corner, I know it's a CD8 T cell because it's CD45 positive, CD3 positive, CD8 positive, CD4 negative. And we can rely on this whole idea of, seeing really is believing. In that you're looking at all of those signals, one of them's black as it should be. This is a very similar conclusion that you might draw, say, from flow cytometry. So it's the ability to positively ID each and every one of your cells by first analyzing them in bulk. What we're kind of bringing to this with our newer instrument, which is the CellScape, is we're able to do this work now at very, very high spatial acuity. We can actually do up to 182 nanometers resolution. So, the nice example I like to pull out here is Ki-67, and kind of the difference that this illustrates is essentially with, anything short of this resolution, you can kind of tell whether a cell is positive or negative. But in this case, we could actually quantify those puncta and actually tell differences between cell populations based on that. So it's not necessarily to say, this is a, a game changer one way or the other. But this I find is very exciting in the ability to actually give somebody the ability to do subcellular phenotyping, subcellular localization, which is pretty cool. And of course, we can also now do, whole pieces of tissue because of the speed of the new instrument, which is pretty cool. So I always kind of like to take a little pause here at the beginning and talk about multiplexing in general and again, spatial biology. I actually worked in Nikon instruments before this. I was a kind of large systems reps, confocal, super resolution, things like that. And one of the last demonstrations I did was the application anyways, was taking 12 different fluorophores on FFPE tissue. And they wanted me to use the spectral detector on my confocal in order to unmix those. In theory, this should be relatively easy, but actually in practice, it was pretty difficult. You needed to have all the reference spectra in order to unmix it. There's lots of caveats to go with that approach. And essentially, I was unable to do it. And, I haven't gotten over it. But the real point of this is really that what I was being asked to do, and again, this was in like 2018, is essentially spatial biology. So this was actually to make sure that the instrument I was selling could be used with CytoMap. So CytoMap's actually been around for quite a while, even before 2018. So the point being that, calling something spatial biology, certainly the term is new, and certainly there's been an explosion in the number of solutions that are available, but the actual work and the actual application is fairly old. And there are people that already have kind of formed opinions on what you should be doing here. So, kind of learning from that, we kind of identified and hopefully implemented maybe kind of three main philosophies here with our approach to spatial biology. The first in the middle there is that there are certainly people that are making giant cell atlases with 150 markers, absolutely. But for the most part, most people that want to look at spatial biology, really what they're interested in is they have one or two markers that they've been working that are probably their career. And what they're looking to do is typically put, say, 15, 20 markers around it to give that context. That's really what they want spatial biology for. So it needs to be flexible. It needs to be able to use those particular markers. What you don't want to do is try to shove somebody into a box, either through your reagent space or through your technology space that disallows them doing that sort of work. And that's really kind of what drives the fundamental philosophy of this way of doing spatial biology is we allow you to do that. The second point here, because the third is multiplexing, it's not new, and I already said that. If you're going to do faster, if you're going to do automation, it can't get in the way of the quality of the data. So this is something we really learned in the development of the CellScape. We actually had a legacy instrument, still do. It's called a Zell scanner. The Zell scanner was essentially a Zeiss microscope with some other stuff attached to the sides of it. But the key there, the Zeiss microscope, it's a very high quality instrument. It collected very high quality data, and it allowed us to do the rest of the analysis work and empowered that analysis work. So in our striving to go faster and striving to add automation, we did have choices that we made, whether it was going to be very fast or it was going to be very high quality data. We always chose very high quality data because in the end, it doesn't matter how fast you produce the data. If it sucks, you can't use it. So if you're going to do multiplexing, especially if you're going to do multiplexing, it has to be very high quality data. Okay. So, there's from that kind of philosophy or from that viewpoint, three things kind of come out of this, or three things that kind of drive what the CellScape is or what ChipCytometry is. The first is this ability to use open source reagents, which we'll get into here. The second is collecting what I call biologically relevant data. So that's that very high quality data set that we can actually use and we'll put up with bivariate that'll put up with cluster analysis. It's good enough to actually give you an answer. The last thing is really open sourcing the data that you get out of this. So we just had a conversation about before this talk, these data are huge. So you need to be able to deal with that in the first place. And you need to be able, again, because people already have an idea of what you should be doing with spatial biology, there's already tools out there. You have to give people the ability to use those tools that they're used to and they're doing other work with. So that's our approach with that as well. Kind of setting up the reagent scheme here, what we're doing. Again, the CellScape runs an assay called ChipCytometry, and ChipCytometry is essentially cyclic immunofluorescence staining. You see on the left here, we truly can do just about anything that you can section, anything that you can pipette onto a cover slip. We try to make this as agnostic as possible so that you can kind of, so it marries well with this idea of being able to use any reagent you want or any antibody that you want. So we can do FFPE, we can do fresh frozen, we can do cell suspensions. Our kind of bread and butter here is typically PBMCs. The other thing I'm very interested in, especially specifically for the IO space, is we can do kind of truly correlative circulating versus resident TILs, which is, I think, very interesting and starting to kind of catch on as one application where you can actually look at the population that's circulating versus the population that might actually be embedded in a tumor and then compare those two together. In order to do that, you need to have reagents that are close enough in antigenicity to make that bridge. Once you're there, though, what we're going to do is multiplex is cycles of 5 here. So if we've got 40 markers, we might do 8 cycles of 5. We're going to stain. We're going to collect through microscopy and image capture. We're going to collect images of those stains, and then we're going to importantly just use photo bleaching in order to erase that signal. So signals erased, so then we can stain and image again. So we go through these cyclic rounds of staining, imaging, photo bleaching, staining, imaging, photo bleaching, staining, imaging, and photo bleaching. And then we collate all those together into one large multi-channel image. So that's the idea. But again, what the kind of photo bleaching allows us to do is we can use a much wider breadth of reagents. What we found is basically, it has to fluoresce, but anything that excites in the UV vis, anything that emits in the UV vis, we got to get those two, and anything that semi-readily bleaches is something that we can do. So we do a lot with primary conjugated antibodies, secondary antibodies, so on and so forth. And even for things that don't bleach so well, again, because we're just using fluorescent reagents, there's lots of other tips and tricks that we can do in order to bring in reagents that maybe don't bleach. But this kind of illustrates the basic point here. So I've got ChipCytometry on the left, just an antibody there, and then what I'm calling proprietary detection on the right. So I'm going to beat up on oligos and chelating agents particularly, because usually that's what we're being compared to. So if you take the case of proprietary detection, and again, there's lots of ways to do this. Here are the ones that are popular. So if I have a vendor conjugation or demanded vendor conjugation, the problem is that if this doesn't work, if this process fails, you can't use that antibody. So again, if your research requires antibody X to work, and a lot of times this is the case, a lot of the what we take in as projects internally is, can you get my antibody to work so that I can use spatial biology with these other 15 markers that are kind of plug and play. But when it doesn't work, you can't use the antibody. And we also have, quite a bit of information at this point that these conjugation strategies aren't 100% or even close to it. They're closer to like 50-50. So you can imagine if I was taking on a spatial biology project and I had a roll of the dice as to whether the marker I was actually interested in would work, that starts to look pretty dicey, especially when you're trying to get, say, a grant committee to agree with you. But as an alternative, maybe we take a look at ChipCytometry here. So ChipCytometry, again, we can use primary conjugated antibodies for a lot of markers. Those are available from vendors. When they're not, though, conjugation kits. So these do have fairly high success rates. We're talking 80-90%. So the technology behind just getting an antibody on, a dye on an antibody is pretty well established. Let's say that doesn't work either. We can always just rely on secondary detection. This is pretty fail-safe. Not 100% fail-safe, though. So even then, we've got things like biotranscriptive linking and all sorts of other fun things that we can do in order to get your antibody onto a signal. The key is that we have many different strategies that you can employ in order to get the signals that you're interested in to actually work with the system. And that makes it really, really powerful for actually getting work done that's meaningful to you. Other things that are kind of goodies that fall out of this. Because we're using the same reagents, not a reagent that's now modified by some conjugation, it makes validation super, super straightforward. So we combine that with the fact that we're only typically doing 5 at a time. And usually you can take your multiplex section and compare it directly to another slice or another section where you've done a single plex, say an IHC or IF, something that's truly gold standard, something that's going to put up with, say, the pathologist that's screaming down your neck about whether you actually have specificity or not. It's really easy to show. The other reason, I always like to show this kind of image at the bottom here. When you don't break it down to those smaller multiplexes, or you're having to rely on some abstraction in order to tell if signal A truly is signal A, you end up with the situation like you see below. So that's the same CRC we're looking at, but each one of those is an immune marker. And especially when you're talking about immune markers, you've got a lot of signal that are packed onto cells that are very, very close next to each other. It is not at all trivial to try to figure out, who's CD3 versus who's CD4 versus who's CD8 here, without some sort of base validation to go with it. So it's not really a trivial problem. This can get, it's real, kind of real life part of doing this sort of thing, or this application. And again, being able to drop it down to a single plex or a dual plex makes it much easier to do this. The next, really kind of what I guess I've been harping on at this point, but it allows you to do what's called, what I call non-canonical markers. But really what we mean here is like, this allows you to do your markers. So this is an example we typically use for this. So this was it's a publication out of Oxford where they're looking at mate cells. It's mucosal associated T cells, invariant T cells, excuse me. But this was actually these 6 markers were a downsampling of say 36, but these really positively ID'd these cells. So they could, take their 36 markers, come up with a marker panel that really made sense. Fairly low plex means fairly high throughput. But what they were able to do with this is really, well, first and foremost, they were able to provide actual in situ data. So these are actually clinical samples. These clinical samples and the ability to show that these mate cells were actually close to a mucosal layer, backed up a lot of other lines of evidence that were showing that these cells actually had some sort of tissue repair function. So basically they did tons and tons of work that showed that this should be the case, but they used ChipCytometry, spatial biology in general. But in this case, they used ChipCytometry to actually show that was happening in patient samples. And that was really kind of the showstopper of the paper. But again, kind of the main point of this is, well, I would expect somebody to have conjugation worked out for their custom conjugation, whatever they're doing for CD8, CD16, that's fine. I really wouldn't expect them to have a V alpha 7.2. I wouldn't expect them to have PLZF. That would be a custom conjugation project, which may or may not fail and is at least going to eat up a bunch of resources. So we can obviate that, which is pretty cool. The last thing, kind of forward thinking, is again, anything that fluoresces is something that we can use. So we can also take a look at things like RNA scope, or being able to use RNA signal in conjunction with IF. So, in the particular case of kind of this mid-plex of 15 to 20, what this is really useful for is in combining it with, looking at markers or looking at moieties that don't necessarily have a good antibody. So, or alternatively, don't have a good localization. Consider like, basically any cytokine. So that cytokine is, if it's intercellular, is just going to be kind of everywhere. Which is fine, but then if you're trying to assign it to particular cells, which is really the work that most people are trying to do with spatial biology in general, it's very hard to, almost impossible to do that. But what you can do is take a look at the RNA that's actually being expressed and tie that directly to particular cells. So when you have a marker that doesn't necessarily have an antibody that makes sense, or the localization doesn't make sense, that's when you can combine it with RNA signals and have something really, really powerful. This kind of ending on a boring note, I do apologize, but the one thing that kind of underlays all this, or maybe might be in the back of your mind, is like, all right, so I'm going to stain a piece of tissue cyclically with 45 antibodies. How long does that take? In this case, it does, in fact, it takes a while. But what we can do for you then is, with the CellScape, the new instrument, we at least automate this for you. So each of these little bays you're looking at on the left can hold up to 5 antibodies. So we can actually, even without touching the instrument, we could do a 75 plex if you chose. There's also a way to do, the manufacturer calls it hot swapping. Sounds pretty cool to me. But essentially, once you get done with the first 7 vials and you're on to vial number 8, you can depressurize the top row, and then you can start adding even more than that. So essentially, you could go ad infinitum. But again, the more markers you look at, the more time, the more investment, so on and so forth. But at least it's a possibility. The major point is, though, that we take whatever staining protocol you're looking at and we make it automated so that you can essentially, leave at 5 after you blow the machine, come the next day, and bada bing, you've got your data. So yes, so kind of the three things it really gives you is, again, this is much easier to validate than most of the spatial biology platforms. We're truly target agnostic. That's really what we're going for with the platform. We want you to be able to do your work. And finally, it does enable multi-omic approaches, be that, the real showstopper everybody's looking for is, of course, mRNA and RNA, but there's other things as well, too. We can look at an endogenous signal, for instance, and compare that to staining. There's lots that you can do with this. Again, it's just as long as it fluoresces, it's something that we can use. The next kind of part of this, again, is this idea of collecting biologically relevant data. So to kind of set this up, this is a fairly straightforward example or simple example, whatever. I thought it was pretty clever, though. But all we're really looking at is a cell smear of PBMCs with CD4 on it. So CD4 on PBMCs actually is trivariate. So you got a lot of cells that don't exhibit any expression of CD4. You've got some cells that are sort of kind of dim. Those are your monocytes. And then you have other cells that are blazing bright, and those are actually your T cells. In order to identify these individual cell types, you actually have to do some extra hardware, fancy technology, not tricks. But we've employed a mechanism of doing this to actually collect that data again in a meaningful way. So the illustration on the right here is thus. So what I've done is I've used my microscope to collect an image that gives me pretty nice contrast, so I can nicely see those cell borders. In order to do this, I've used an exposure, a camera exposure of about, say, 100 milliseconds. So nice looking picture. But what happens if I increase that to 200 milliseconds of exposure? What you see here is what that arrow is actually indicating is you see another cell show up. So the longer I expose, the dimmer signal I can capture. If we do that again, suddenly I'm seeing all sorts of cells that are actually in that sample. But something else happens when I start to get really, really long exposures, which is I overexpose the bright cells. So I do this again, and you can see, our bright white indicates saturation, but you can see all the other data that's actually available here. So as it turns out, which is a lovely phrase I love when academics use to, let's skip to the end. But as it turns out, that there is most markers, I would say, actually exhibit a signal noise range or an expression value that's outside of what you can capture in any particular camera exposure. So it doesn't really matter how sensitive your camera is, how much you paid for it. It's really the exposure that sets a bracket on what expression value that you can capture. The only way out of that paradigm is to collect multiple exposures. And that's exactly what we do. So we use HDR. So this is the same thing that your camera phone does, but we kind of flip that on its head. Instead of compressing that into one image and going for contrast, that's really what your phone does. Instead, we're actually summating all those values together and normalizing it such that we can look at one large data set that actually covers the actual signal noise that you see within the biological sample. So essentially, we can integrate all the data that you would catch if you took a 4 millisecond exposure all the way up to a 4,000 millisecond exposure. We can really bracket the expression values that you're getting as opposed to just what the camera's giving. Where the rubber kind of meets the road here is, again, when you compare it to data you might get out of a flow cytometry experiment. On the right, so this is some competitor data that was collected on one of their systems. And you can see exactly what's going on here with CD8 versus CD3, where you're actually missing an entire population here of our CD8 positive T cells or cytotoxic T cells, simply because they didn't take short enough exposures or long enough exposures, depending on what they were going for here, but they didn't take the full bracketing series in order to actually catch these cells. So when you don't take this approach of HDR imaging, you actually miss out on data. And even if you're doing this by cluster analysis as opposed to by variegating, what you end up with when you don't collect those outside or those larger expression values or smaller expression values is all your data tends to collapse in the middle. And it becomes very difficult to analyze this one way or the other. It doesn't really matter what your approach is if you don't take this HDR approach. The other thing we're adding to this is, again, just very, very high now spatial acuity. So this is with our newer instrument or with the CellScape. What you see is, most spatial biology applications stop around 500 nanometers per pixel. which as you can see is, it's fine. So you can still make out kind of cell borders here. You can kind of assign roughly who goes or what signal belongs to what cell. But again, we take this actually down to 182 nanometers per pixel. What I'm trying to depict here is, over and above just being able to tell where a particular signal is, you're actually also able to identify here artifacts, things like that, that you wouldn't necessarily be able to see in the other data set. So here I'm just showing this kind of break in the membrane that you can catch at 182 nanometers. So the ability in general just to collect at a much higher spatial resolution, again, just empowers higher and higher, better and better anyways, image segmentation. So we actually had, in the development of this instrument, we had this interesting phenomenon where the data at 500 nanometers per pixel, when we quantified that, the average that we get for an expression of the marker was much higher than we saw when we collected this with CellScape. Shouldn't have been the case, right? We've got better cameras and all this other jazz. What we found out though is that in our analysis, we didn't compensate for the fact that now we can get a much more exquisite border around that particular cell. And because of that, we were actually cutting off the signal that was being averaged by the older algorithm just because it was taking bigger pixels. So we compensated for it. But what it does prove though is that you absolutely can get better segmentation with higher spatial acuity collection. This will empower new science. And again, kind of bringing it back to that example I saw before, this is really, again, kind of an A versus B of what you get on the right at 500 nanometers of resolution for, in this case, it's FOXP3 versus 182. So again, on the one side, we can certainly tell if a cell is positive for that marker. On the other, we can actually count those puncta and we can quantify not just based on the intensity of the overall signal, but rather the distribution and the number of those individual puncta. So that in itself could be really powerful. So I think this is a very good use case that's easy to see that really illustrates all the additional data that we're going to be able to get out of this higher accuracy, I guess, or acuity optical train. The other thing to kind of point out too is, getting higher resolution is not, relatively speaking, like magical, especially for microscopy. Go up in higher magnification, you're getting it at higher resolution, to a point, of course. What's cool is we've been able to, with other changes in the optical train, actually maintain kind of the speed that you get with a low magnification, but still give you the spatial acuity. So we're actually able to give you that 182 nanometers of resolution, but our sampling field of view is still over, it's at 0.8 millimeters squared. That's darn near an entire ChipCytometry experiment in one go. So we can very easily do surveys and collect entire images, but still give you the massive spatial acuity as well as the HDR imaging. So we're really trying to give it all to you. As far as kind of other, again, less boring stuff, but still very important. We've also just increased the size of the window. So now we have over 3 centimeters of window to work with. This will cover, most mid-sized biopsies. Not, we've still got a way to go. And this is still something that we're working on and have some things to announce by the end of the year. But even here and now, we've got a pretty good window. And again, we can do up to 4 samples at a time. So I personally think this is pretty nifty in that they set up this fluidics unit so that it can actually run 4 independent experiments at a time. So I know a lot of you guys are from cores. You could have 4 different PIs running 4 different experiments at one time on your system. Or alternatively, if you're doing a assay development, this could be 4 different dilutions or 4 different combinations of markers. They're truly independent one from the other. So it's not just about faster turnaround time. It's actually able to get through those types of experiments faster as well. The last thing, again, to talk about is this idea of our data pipeline. So kind of soup to nuts, as they say here. So what we're doing is for each of these markers, we're taking HDR series, and then we're going to collate that into a single HDR image that we're going to analyze. We find those individual cells using cell segmentation. We apply the background signal first in order to get true quantitative data. Montage those guys together so that you're looking at a piece of tissue as opposed to individual fields of view. And then we do our segmentation. Currently, that's based on Stardust. So kind of the latest and greatest we found at the time. Then there's a series of bivariate gating within our software that's done manually, and that'll give you populations. But really what I would say is, this again is kind of the onboard out of the box analysis that'll get you going. You install it on a Monday, you'll be able to do this by Thursday. It's certainly not the end all be all to what people are actually looking for as far as these data streams. So this will get you started. And I would say at first, this tends to satisfy 80% of customers. In fact, we still have people that are just doing this population-based analysis and have been doing it for 6 or 7 years now. But for those that are not satisfied with that, and I would say that's academia, that is research, we do have the ability to offboard these or export this data and then make it easy to import into other people's software. We basically found that there's kind of 3 approaches that people typically take here. The first is kind of the open source ImageJ QPath skill set, as well as kind of philosophy, where really all you want is the data and you want to be able to use your own pipeline. So for those people, the way we support that is just having a very open source and standardized file format. So there isn't the Canopy file format, it's just OMI-TIFFs. We also then supplement that with our guys and gals, of course, our technical aides being able to support you in using that, at least to get the data into those programs. I'd say the second tier here for people that are willing to pay for software and want it to be easy use and things like that. You have your solutions like Halo and like PhysioPharm, things of those nature. So for that, the support's not really nearly as much on us. They have support for you, but you still need to be able to have an OMI compatible TIFF. So the way that we support those customers is we have partnerships with those vendors. We do posters with those vendors and things like that, and also that OMI TIFF format. We lately identified, because of the size of this data, and not really just ours, but really spatial biology data, there's kind of a third kind of use case there, and that's people that are doing this on a truly kind of enterprise scale. If somebody's not looking at 4 samples, if they're looking at 400 samples, you actually do need specialized computer hardware in order to get through that by the time your PhD student graduates or you have to move as a PI. So we have solutions for that too. So that specifically, we partner with a company called Enable Medicine. They specialize in cloud computing so that they actually can, and they showed us, and it's really cool. They can scale, because it's all cloud software, cloud compute. They can do 4 samples in the same amount of time that they can do 400. You just crank up the number of virtual machines that are working on the problem. So that's kind of those kind of 3 scales or 3 approaches that we try to support. And we find that really does satisfy most people. Just some examples here. This is a paper out of the Bush Lab, Technical University of Munich, where essentially they have taken that first approach where we collected the data on our machine, but then the entire analysis pipeline was actually built from scratch in ImageJ. Also using, I think he used self-poses segmentation. But he got a very, very nicely and exquisitely kind of differentiated population for his sample. But again, what I find here is when you try to implement this as a single lab, there are some caveats to it that made it very good for his work, but not necessarily good for everybody's work. So again, it's really not that this is the approach that you should take. It's rather that this is the approach you should be able to take with our data, and we can do that for you. Same deal here. I think we're just showing segmentation and QPath just directly with our image pipeline. QPath, if you're not familiar, is really great for this kind of work. I'd recommend it for spatial biology, whether you're using ours or anybody else's. It's a really nice, easy pipeline that's still kind of extendable, and it's still freeware, which is great. So again, kind of summing up, I skipped my slide. Apologize there. So kind of summing up, so we're collecting data again. That's very we're giving you the ability anyways to collect data that's actually meaningful to you, meaning that you can collect from markers that you're interested in and build up a panel that you're interested in without any compromise. And we're doing that with high enough data integrity, whatever you want to call that, in order to use bivariate gating. So in order to use manual bivariate gating, you have to have a very large signal to noise so that a person can actually identify those individual populations. We can still do cluster analysis and things like that. But this is really kind of, I would call it the gold standard for this sort of work. And then finally, we have an onboard pipeline that'll give you populations. But if you want to go beyond that, we certainly have the entire tool set in order to be able to do your neighborhood analysis, neighborhood classification. And honestly, what I'm interested in is whatever cool thing comes out this fall at SBI2, or all the cool tools that I'm sure are coming in 2024, 2025, and so on. Cool. I believe with that is my time, and I really appreciate your patience. And if there are any questions, I'm happy to discuss. Thank you very much. Questions on the chat?
I had one on the optimization of antibodies.
Mm-hmm.
Sometimes the order they're put on.
Sure.
Make a difference. How do you guys go about that?
So we tend to so we have the benefit actually of there. Yeah, of course. So the question is about assay validation in order of markers, I would say. What is our process for kind of identifying what the optimal order is? That'd be fair. All right, cool. Mm-hmm. Okay. Mm-hmm. I assume my licensing fee for my likeness we're good there. All right. Good. Okay.
There you go.
All right. Maybe I'll business this way. All right. Sure. Yeah. So the regarding kind of putting together these assays or putting together these large lists of markers, because we're doing it cyclically, are there order of operations issues? And there absolutely are. What we found though is that for the most part, it has everything to kind of do with the epitopes. Some epitopes are just, call them fragile versus robust, but some of the epitopes just need to be at the beginning of the assay. We found that to be the major issue or bottleneck, whatever you want to call it. So CD19 is particularly is a real bear. If it's not in one of the first rounds when we do it, you won't be able to catch it. But we know these things, and we have kind of the internal training as well as the kind of the technical help in order to walk you through it. Generally, if we're looking for proof positive that we have an assay and we're happy with it, we'll run that assay forwards as well as backwards and just look for differences in the expression values that we get. Usually, if there's a major issue, you'll catch it there. The other thing that's cool about doing it kind of cyclically is at every one of those rounds, if you catch one of those issues, you can look at it then and there on that round, as opposed to trying to decipher that from 39 other markers if it's a 40 marker panel. So that's how we tend to approach it. Yeah, absolutely.
So actually, a question did come in.
Cool.
The chat.
Mm-hmm.
So this is from Dana King. She says, really nice introduction to the technology and platform. Thank you. Regarding the analysis, one, are multiple exposure times used for quantitative results? And two, if so, is variance stabilization or a similar normalization steps recommended? Used to account for the higher dynamic range expected in the question of data.
Mm-hmm. Sure. Yeah. So HDR imaging is used for the quantitative analysis. So it is HDR imaging that we're analyzing in order to do this. We've, in the development, we've looked in the type variants. What we've kind of landed on, and I don't know if this is necessarily because on our development side, which is also the CRO side, they've been doing this since like 2009. So for the most part, nowadays anyways, what they're looking for is for co-localization with markers, and then also just secondary or specific staining in general. So, one of the things that higher signal noise allows you to do is you can actually look at higher concentrations, lower dilutions, however you want to look at that. So because of that, we can often catch, very, very dim signal as very, very bright signal. And what that really allows you to do is that your fine tuning, I guess, that's required on that dilution. You suddenly got a whole lot more slack to build those assays than you did before. Because you can imagine, if 1 to 100 gives me a particular signal that's then tied to a particular exposure, if I screw that up very much by, too dilute or too concentrated, I'm going to either be oversaturated or I'm not going to catch the signal at all. We've got the benefit of actually having, because we have that higher signal to noise, if it's a little bit dimmer, we can still catch that. If it's a little bit brighter, we can still catch that. And of course, if it's a lot dimmer and a lot brighter, we can catch that too. So it gives you a little bit more design leeway with the assay and putting those assays together. But we don't necessarily do as many variant controls anymore unless we're asked to. A lot of both clinicians as well as big pharma just ask us to do those things because they need those tests to be done. But for the most part, you can tell whether you got a good assay or not, just looking at co-localization with markers, both negative and positive on particular cells, as well as just looking for, just general non-specific antibody binding, which is usually pretty telltale. I hope that answers it. If not, yeah. Maybe another thing to mention now is we do have we have some space here next to the auditorium if you'd like to meet with us anytime this afternoon. Additionally, definitely feel free to reach out to Mike or myself if you have additional questions or if I'm not actually answering your questions well enough.
I'm going to also ask, you said I think the FOV is like 0.8 millimeters squared,
Mhm.
right? So how long does that take to scan?
It can be really, really fast or really slow if you are taking a lot of markers. So,
One set of 5.
oh, 1 set of 5, like, I don't know, seconds, something like that.
So
Less.
you can probably, so you do like, I think you said your region has a 3 centimeters.
Yeah.
So
Yeah,
how long
3
does it
centimeters
scan?
square.
Just like 5.
I don't know if we've done like a land speed test lately, but I would imagine if it's 15 minutes, I'd be surprised. But I'd rather get back to you with solid
Yeah,
numbers.
not
I know
like-
somebody's
so
collected it.
It's not like if you had 40 markers, you could still be able to do that in a day or a night.
Yeah. I think our general now with the metrics we're happy with sharing at this point, meaning they're not too ridiculous and then also underselling. Is we can do 20-25 marker assay on 4 samples in a day.
What about registration between scans?
Yeah, we actually, so because we use photo bleaching, we can't use a fluorescence image for that, or at least we don't. So we actually use a bright field image and register using that. So in process, the thing is aiming the field of view correctly. Once it's got it, then it switches over to fluorescence and takes the images. So it should be perfectly registered every single time. Yeah, absolutely. That actually, the other thing that kind of builds off that is, with the sample in place like that, that's not a challenge really. Most microscopes can do that. The challenge is like if you pull that off and put it back on again, which we actually can do with, we can do a reinterrogation experiment, is what we call them. But when you try to re-register that, then that actually becomes a little bit of a bear, and that's something that we can do too. Yeah.
One of the questions about the 4 and sample of independent managed, are they all being run on the same panel, etc.?
They can be 4 separate panels if you want. The only real, I guess, the limit would be if you wanted to be totally walk away, you would have to divvy up those 15 positions by those 4. But yeah, it's the software anyways, on the software side, it's independently figuring out who goes what, and it's all based on the sample. Cool.
No other ones online? Thanks.
All right.
Thank you so much.
Cool. Yeah. Thank you very much for your time. Good afternoon.
Thank you.
Mhm.